All right, so the goal of this experiment is to understand paper chromatography. Paper chromatography, well, a Russian scientist was the guy who developed the paper chromatography, actually chromatography in general, in the early 1900s. And what he did was he separated the coloring of plants, the green coloring of plants. This technique is really important when we want to analyze many kind of chemicals or biological specimens. For example, we can use this technique to do DNA analysis, or even it helped us identify some victims when we had the 9/11 attack on the World Trade Center. What chromatography does is it separates a gas or a solution, liquid solution, by passing them through a porous material such as ceramic powder or paper, thin film, or plastic sheet, etc. While some of these procedures require really complex equipment, paper chromatography can be done in a very simple experiment. You start off with paper, and what's going to happen is we're going to put this in water, and the water is a homogeneous mixture. So what happens is the water will suck up go up the paper through capillary action. And a homogeneous mixture is a mixture of two or more substances. And so as the solution passes through the paper, the solute separates due to their interaction with the paper. Now, they're going to separate by their molecule size or even their molecule mass. The larger, more massive molecules will be kind of not move as far and left behind, whereas the smaller and less massive molecules will diffuse up the paper further. So let's see what they have. All right, what we're going to do is we're going to take some copper nitrate and some lead nitrate, and we're going to put it inside of a bin. Oh, first thing they want you to do is set up the kind of guide for this. Now, what I'm going to do is I'm just going to take a paper and I'm going to mark off 1 centimeter from the edge. Now I'm going to put a dot at 3 centimeters, 5 centimeters, 7 centimeters, and 9 centimeters. So there. You got 1 centimeter from the edge, and then you go every 2 centimeters spacing in between, so that way you have 5 dots. The lead in here is insoluble in water, so it's really just going to act as a guide, and it's not really going to affect the experiment as much. Now what they want is 3 drops of copper nitrate, so that would be this guy, and 3 drops of lead nitrate, so that's 1, 2, 3. Let's go with 3 drops of lead nitrate. So that would be 1, 2, 3. Oh, almost got 4 there. And we want to mix this with a small mini pipette. Use the mini pipette to put a drop of mixture on the first mark. So I'm going to mix it up a little bit like that. And then I'm going to take this, suck it up, and just put 1 drop there. 1 drop. Got it. Now I'm going to let that dry a little bit. And while this thing is drying, I'm going to put some other marks on there. So let's see. Put a dot of that on there. We'll try other things. I only brought markers with me, but you can try things like fruit juice or food coloring or other felt tip pens or vegetable juice, that kind of stuff. And the different colors actually make some interesting selections. Let's just go with the yellow here. I don't have a black felt tip marker upstairs with me today. And I'll put another one right here. Now, what I'm going to do is I need another drop of that stuff over here. So I'm going to put another drop. There's another drop. It needs to dry, so I need to wait for this to dry. Since I'm just doing a demo video, I'm going to cheat a little bit. There's my quick dry, and I'm going to put another drop. You want to repeat this about 4 or 5 times. Now I'm not going to do this all 4 times. because, again, this is a demo. And what we're going to do is we're going to take this, roll this up into a little tube, and staple it. Now, the reason why I'm stapling it is because the metal is not going to interact with the water. So, again, this isn't going to mess with my experiment. If I can even get it stapled. A second. There we go. And voila. Okay. I'll do another one over here. Almost. There we go. And I'll do one more right in the middle, just to kind of add stability. There we go. I've got my stapled tube of paper. Now I'm going to take my 150 milliliter flask or cylinder and my beaker, and I'm going to pour in about 3 milliliters of distilled water. So here's some distilled water. This is at about 4. Let me just make it about 3 to 4. All right, that's about 5. Okay, that's good. All right, so I'm going to pour it in here. So not a whole lot. It's just enough to kind of coat the bottom of the beaker. And what I'm going to do is I'm going to put this paper cylinder into the beaker with the spot side down. So I'm going to put this inside. There we go. Now remove the beaker when it has diffused. When the water has diffused to the 5 millimeters from the top of the edge. So this thing is going to take a while. Actually, it's starting to work a little bit. You can kind of see, where's the camera? Right there. You can kind of see the water is pulling up this thing, and that kind of pulls up these spots. Now, this first mark right here is right next to my staple, and that's the what was it, the lead nitrate and the copper nitrate mixture. And what we're going to have to do is, this thing will take about 30 minutes. So that thing's going to go all the way up. So we're going to set that aside and wait 30 minutes. Although I'm not going to wait the full 30 minutes because, again, this is just the experiment. But I'm going to kind of tell you what's happening here. After this thing is done, we're going to need to use the potassium iodide to kind of develop the first dot that we had, the copper nitrate and the lead nitrate. And I think what you do is you put a few drops of potassium iodide in a small beaker and lightly wet the cotton swab with this solution. Okay, so we're going to put a few drops of this thing in here and lightly wet this thing with the potassium iodide, and then we'll kind of use that to swab the first dot that we did, and that will make it so that we'll be able to see it, and it will develop or appear. So that's how you do the lab. Then what you want to do is you want to measure the average distance that each solute has traveled and record that distance in millimeters in the table provided. Now, you're might see the dots stay there and then move completely, or you might see the dots kind of just streak all the way up, depending on what you use, of course. Now it looks like all of these dots, you see, they started off at that small dot right there, but then they're starting to move up as this thing uses its capillary action to pull that water up the streak. And what you want to do is you want to measure it. Now, some of you are going to ask me, well, where do I measure from? Do I measure the top of the dot to where the streak is? Or do I measure the bottom of the dot to where the streak is? Or do I measure like the center? And my answer is going to be this. That's for you to decide. But I would recommend this. As long as you stay consistent. For example, if you decide to measure the bottom of the dot, you see how this dot is really not a point, it's kind of a long streak. If you decide to measure from the bottom to that spot, then you're going to have to do that same kind of measurement from the bottom on each of them. But maybe the bottom might not be clear to you, or maybe the top might not be clear to you. Maybe you want to find an average. Well, you make sure to do an average for all of them. Because I actually don't think that it says which one, where to do it from. So I'll let you folks decide on what you want to do. And you want to record that in a table and make sure you answer the questions. And then from that, give me a full lab report. I hope that helps.